So welcome again. We'll go ahead and discuss what we've been discussing. So in the earlier session, we talked about the neurotube and the various neuropores and the time that they usually close and open, and the time that they usually close. We also talked about the neurotube derivatives. So at this point, we are going to start now with the neurotube derivatives. So for the neurotube derivatives, we can just start with this. So this neurotube is undifferentiated. And then you want this neurotube to give you the brain, you want it to give you the cerebrum, the cerebellum, the brainstem, and the spinal cord, the cranial nerves, and the spinal nerves, and everything else. So it has to differentiate. So the cranial neurotube at this point will give us the primary brain vesicles. And also it will also give us the secondary brain vesicles. That is now for the cranial neurotube, the one that is coming from the spinal cord. So, and then the other thing is that you need to appreciate now the secondary neurotube, the structure that will come from the secondary neurotube will come from, will give rise the spinal cord. Yeah, so the three brain vesicles, the three primary brain vesicles that will come from the cranial neurotube will be the prosencephalon, that is the forebrain, the mesencephalon from the midbrain, that is the midbrain, and then the rhombencephalon, that is the hindbrain. And then now, so those one will be coming from the cranial neurotube. Then for the other derivatives at this point, they'll be coming from, the secondary neurotube will give us the caudal part, the distal portion now, the sacral region of the spinal cord will be coming from the secondary neurotube. But now that means the lumbar spinal cord, the cervical, the thoracic will be coming from the primary neurotube. Yeah. So some of the derivatives of the neurotube, but now today we'll be talking about spinal cord development. So at this point, for this spinal cord development, we need to appreciate some few structures. But okay. So let me try to see how we can help the people who want to join. Yeah, but just a minute. So at this point we are talking about the spinal cord coming from neural from the neurotube. So spinal cord ideally you have the process of primary neurulation and secondary neurulation happening to give you the neurotube. Then what will happen is that you'll have now the differentiation of the caudal neurotube to give you the spinal cord. Then the remaining point that you'll have positional changes happening at various ages, and then you'll have the relatable congenital anomalies and embryological basis. So that is the scope of spinal cord development that we're going to cover today. But I would like us to maybe pay a keen interest on what is going to happen in the next few slides, given that will form a major base of our discussion today. So at this point, I just want us to imagine you have your neurotube, then you have a transverse cross-section of this neurotube. So you have your surface ectoderm, here you have your neural cells here, then you have your neurotube here, then you have your notochord. So at this point, you're having your mesoderm, the mesenchyme here, and then of course we'll be having your endoderm at this point, because it's a transverse cross-section. So these cells, you will have an epithelium. This is known as neuroepithelium. So this neuroepithelium is pseudostratified columnar epithelium. So what will happen is that you'll have these cells differentiating to neuroblast and neuroglial cells. And maybe I can pose a question to us. What are some of the neuroglial cells that you know that are found in the central nervous system? Some of the neuroglial cells that you know that are found in the central nervous system. So what will happen is that we'll have pseudostratified columnar epithelium, this epithelium here, which is this, undergoing proliferation. It will proliferate to give us three layers. So these are neurotube being formed, these are folding, the folds layer later. So these are not called neurotube formation. So we'll have the neuroepithelium. We'll have the neuroepithelium undergoing proliferation to give three zones. So we have zonation of the neural tube. So we have the neuroepithelium undergoing proliferation to give you three zones. One of them will have the layer that is just next to this neuroepithelium, which is known as the ventricular zone. It's called the ventricular zone because it's just found next to the ventricles, to the canals, to the central canal. So you find this neuroepithelium will be the one that will now form the ependymal cells that will continue to produce cerebrospinal fluid. Then after that, you'll have an intermediate layer. This intermediate layer here will be called the mantle zone. The mantle zone is the intermediate layer. Then you have an outer zone. An outer zone is known as the marginal zone. Yeah, so it's called marginal zone because it's actually like found on the margins, so on the outer layers. So on the next slide, so I presume at this point you've already gotten the three zones. An interesting thing will happen for this mantle layer. So for the mantle layer, it will undergo differentiation also. And this one you can be brought in an exam setting as a spot question. So what will happen to the mantle layer is that it will undergo like segmentation somehow. So you'll have the marginal layer being intact, the ventricular layer being intact. But now this mantle layer will form the alar plate and the basal plate. So the alar plate is a sensory part of the neurotube, and then the basal plate will form the motor part of the neurotube. In addition to that, now the alar plate and the basal plate will be separated by sulcus limitans. Sulcus limitans will be separating the alar plate from the basal plate. And then maybe to finalize that, you'll find that this roof plate will be now supporting the neurotube proximally or on the on the dorsal points. So you can appreciate this is the dorsal point. And then on the ventral cord, you'll have the floor plate. So this one will be supporting structures. So you can be told in an exam setting, draw a differentiated neurotube. So what you can do is you can draw this. So you can draw the ventricular layer, you can draw now the mantle layer having the alar plate and the basal plates. And then you can also draw the marginal layer. So point of appreciation is that the basal plate will give us the motor derivatives of the spinal cord, of the brain, of the cerebellum. All those motor derivatives will be coming from the basal plate. Then the alar plate will give us all the sensory derivatives, the spinal cord, of the cerebrum, everything that is sensory will be coming from the alar plate. Anything that is motor will be coming from the basal plate. And that's the reason why the sensory root of the spinal nerve ideally comes to the alar plate on the dorsal aspect, and then the motor root of the spinal cord of the spinal nerve usually comes from the ventral aspect, which that means from the basal plate embryologically. So just to show us the derivatives of the mantle layer, you can appreciate the alar plate, the basal plate. So the basal plate will give us the ventral horn, and this ventral horn is motor in nature. So we can actually appreciate its ventral motor horn. Then the alar plate will give us now the sensory or the dorsal horn. Then another point of appreciation is that this the white matter will be coming from the marginal layer. So the marginal layer ideally axons, fiber tracts. Then for the central canal, ideally that one remains entirely in the spinal cord, in the brain, give us the ventricles, the central canals, the cerebral aqueduct, but also maybe to familiar to make us appreciate that this ventral horn and the sensory horn are ideally at this point, they form the various nuclei. So remember, we had a dorsal root ganglion in the peripheral nervous system. Then this collection of cell bodies in the spinal cord, in the brain, they will be known as nuclei. Ideally, the gray matter at this point. The various nuclei will be found in the gray matter. So the basal horn, the basal plate and the alar plate will give us the gray matter. But now this gray matter for the basal plate will be motor, and then for the alar plate it will be sensory. I hope that one tries to make some sense in a way. Then you'll have developmental changes of the spinal cord levels. So I presume this one you covered during your lab session. So maybe someone can tell me what is the spinal cord level at 3 months of intrauterine life. You can put in the chat box, or maybe you can just arrange it for us. At the third month of intrauterine life, at the sixth month of intrauterine life, at birth, and then at puberty, and then at adult. What are the various positional changes of the spinal cord level? Anyone who would like to try at birth, or maybe at 3 months intrauterine life? Yeah, at coccyx, correct. You're right. So Katrina, and if you're correct, it will be at the coccyx. What about 6 months? 6 months. 6 months. S1, S2. Ideally should be S1. Should be S1. What about now at birth now? Because at now you can just figure it out. At birth you have L3. What about puberty? L2. What about now adult at adult stage? Um, yeah, Jonathan, I can see where you're headed. It's L1 ideally, but no, it's actually between the border of L1 and L2. The border between L1 and L2. Yeah. So those are the various positional changes. These are the various positions. So I did just the way you mentioned about it. So 3 months intrauterine life, it's coccyx. 6 months intrauterine life, it's ideally at S1. At birth, it's L3. At puberty, L2. Adult between L1 and L2. Yeah. So maybe someone can just tell us, why do you have those positional changes? Why do you have those positional changes in the spinal cord levels? So at this one, actually, you can actually appreciate this one is at S1. So this one is 3 months intrauterine life. You can appreciate now, sorry, at this point, you can actually appreciate like this, the whole of the spinal cord is actually at the coccyx. The whole of the spinal cord is actually at the coccyx. Then as time goes by, like it's at now it's actually at S1. So this one is now 3 months in the uterine life. Then currently appreciate now at this point it's actually at L3. So this one tells you it's at birth. Then you go ahead, puberty between at L2, then now at adult in between now at this point. Yeah. So the reason why you have, yeah, exactly, the vertebral column grows faster than the spinal cord. So CM, you're correct. The the vertebral column develops faster than spinal cord. So you can be told, explain the various developmental changes that happens in the spinal cord. Yeah. So that is it. Then we can go ahead and now talk about the various congenital anomalies. You have neurotube defects. There are many. Then you have rachis, you have syringomelia, and then you have tethered spinal cord. So for neurotube defects, I know you've seen some of them: spina bifida cystica, spina bifida meningocele, myelomeningocele. Yeah, and then you'll have now rachis, split spinal cord, syringomelia, presence of cyst in the spinal cord, and then you'll have tethered spinal cord. So this tethered spinal cord, that means it can be able to have those developmental changes that we talked about. So we have various types of neurotube defects. So at this point, you can appreciate this is a normal spine. So in this spine, you can have two types of neurotube defects, like broadly, two types. And kindly hear me out in this. You have two broad classification of neurotube defects. One of them is spina bifida occulta. Then if you see a cyst, that is spina bifida cystic. So we have two broad categories of neurotube defects, spina bifida occulta. So this spina bifida occulta is where you see now the tuft of hair, as you'll see in the next slide. Then for the other one is, if you see a cyst, for example, in this one, you can appreciate presence of a cyst. So spina bifida cystica. Presence of a cyst, spina bifida cystica. Now, so that if you just see a cyst, like you can't be able to tell what are the contents of this cyst. Then if you can be able now to tell the types of cysts, if you know you can be able to see the contents in this cyst. For example, now in this one, you can appreciate like you have just the meninges, but absence of nervous tissue. So absence of spinal nerves, absence of the neurotube. That one is spina bifida with meningocele. So or spina bifida meningocele. Then if you can be able to appreciate presence of nervous tissue that is at the meninges now, and that will give spina bifida myelomeningocele or spina bifida with myelomeningocele. So myelo means nervous, and then meningo means meninges. So spina bifida cystica will be further subdivided into spina bifida meningocele, spina bifida myelomeningocele. So I hope you've gotten my point. You have two broad classifications: spina bifida occulta, spina bifida cystica. Cystica is further divided into spina bifida meningocele and spina bifida myelomeningocele. So in myelomeningocele, you can see a nervous tissue. In meningocele, just meninges and the cerebrospinal fluid. I hope that makes sense. You can go next to our next slide. So, but before then, you'll appreciate why will you have now this cyst coming across? It will come across due to now a defective formation of vertebral column. So, you'll appreciate now a vertebral column at that point will be forming not well. So this one is now a good definition of spina bifida character. You can see a tuft of hair at this point. So this one is spina bifida character. So in the same case, tuft of hair, spina bifida character. And actually, there are so many myths. In India, they usually say that a person with spina bifida character is someone like a spiritual person. Yet it's just an anomaly. Crossed fingers. Anyway, on this other end, you can appreciate spina bifida cystica. So this one is spina bifida cystica because you can't be able to say the contents in this cyst. Then for this cystica now, if you can be able now to see that like the presence of absence of a nervous tissue in this cyst, that is meningocele, presence of a nervous tissue in it, that is meningocele, meningomyelocele or myelomeningocele, can be either way. So I hope that makes sense. So this one now is rachis. So that means a split spinal cord. A split spinal cord. So rachis is a split spinal cord. So that means the vertebral column didn't form entirely, like a better component of it didn't form. So you'll find that the neurotube, ideally, it will fold, but ideally this spinal cord will be absent, and then you have a split spinal cord. So maybe I can just tell someone to tell me, or maybe someone can just tell me. So when you have now the brain and the spinal cord being like being open, so you have absence of formation of vertebral column, absence of formation of the neurotube. What is normally given to that um, syndrome? Anyone can just, yeah, craniorachis is correct. Craniorachis, you're correct. So craniorachis, that means the cranium and the rachi, ideally at this point, is the vertebral column. So all of them are affected. What about this? Is this craniorachis? Is this rachis? Is it cranial? Yeah, this is rachis correct. This is rachis. Just actually appreciate is a nervous tissue. The spinal cord is just open. Then you can appreciate that. So for the other one now, the other one now, this one is now cranial rachis. You can appreciate the spinal cord and the brain is open. Then this cranial rachis. So it doesn't come alone. It comes with now anomalies. Remember, these anomalies are usually these anomalies usually come as a spectrum of anomalies. So you can't find craniorachisis at all. So you'll find that person maybe has pharyngeal arc anomalies, craniorachisis, acalvaria, so many anomalies coming across. Yeah, so then you'll have now syringomelia. Syringomelia is now presence of a cyst in the nervous tissue, as you can see in this. So this is the neck region, the cervical region. Then you have now this is a nervous tissue. Then you have cyst filled with cerebrospinal fluid in the spinal cord. So presence of a cyst. So syringo means cyst. And then really, of course, now nervous tissue, presence of a cyst in the nervous tissue. Then at the time you can also you can see presence of a syrinx, presence of a cyst in the spinal cord. So that is also syringomelia. Then maybe to finish up, you can appreciate now tethered cord. Tethered cord. So that means the normal spinal cord is supposed to have migrated that way to L1. But now it's tethered, so that means it can't be able to move. Causes of this tethering, it can be maybe now this defective uh, pia mater, the filum terminale. It was replaced with a fatty tissue, or maybe there was developmental uh, retardation that was happening. So for these anomalies, then ideally for a person with craniorachisis, rachisis, like for rachisis, chances of survival are high than in craniorachisis. But ideally there is no treatment, given this is nervous tissue, there is no treatment, maybe supportive care. And not zero treatment, maybe that's where we come in. You can try to come up with new technologies and try to treat them, or maybe new medications to treat them, but ideally there's no treatment as per now. But of course, there is prevention by taking folic acid. So that's the reason why folic acid is important for the process of neurulation. Even uh, for the process of formation of the nervous system, folic acid is recommended. Just enables myelination and everything to happen at this point. Yeah, so any question? Any question? So someone is asking, is the same mechanism of formation of the spinal cord the same as that formation of the brain? Like the ala, the base of place. Exactly. So now we'll be talking about development of the cerebrum and the and the cerebellum and the brainstem. What will happen is that you just have maybe excess folding or less folding. So remember the three primary brain vesicles that we talked about, the prosencephalon, the mesencephalon, and the thromboencephalon. So those will give us the brains, the higher centers, the central nervous system. So what will happen is that now the basal plate will give us the motor component of the cerebrum, the cerebellum, and everything. Then the ala plate will give us the sensory components of the cerebellum, cerebrum, and the brainstem. But we'll cover that maybe on a later date. I hope that tries to answer the question. So what happens is that I just have a different modification happening. Yeah. But that's a good question. That's a good question. So these are sample essay questions that you can have, and you can be told state the embryonic origins of the nervous system. You can be told give an account in neurulation process, 8 marks, list neurocrest derivatives. Can also be told name the zones of the distal neurotube and state the derivatives of each. So this one is now the one that I was giving you now, the diagram that I showed you. Then state bits of spinal cord termination at L1, L2 junction in adults. So you state the basis of it, and then of course you're supposed to go ahead and talk about the types of neurotic defects. So I presume by now you can be able to answer those questions and come up with good answers. Ideally, that is what you can be asking in neurulation. For our various anomalies, we'll be asked craniorachis, rachis, spina bifida anomalies. You'll be asked all of them. Yeah. So just be prepared. So at this point, maybe I can take maybe some few questions here and there before we end the session. Anyone with questions or maybe clarification that you'd like to ask, you can ask at this point. Any questions? You guys are good? How differentiate chordomas and syringomelia? Yeah. So for chordomas, okay, we have first of all we have two types of chordomas. Maybe someone can just tell me what are the two types of chordomas. So remember, for syringomelia, it's presence of a cyst in the nervous tissue. Then it is actually not big. It's actually a very small cyst. For chordomas, they are big. Yeah, they are big. Yeah. So we have various types of chordomas. I want someone to tell me what are the various types of chordomas. Yeah, two. And then chordomas, where do they come from? Nope, they're not cranial and spinal. Uh, so you can call them cranial and spinal somehow, but ideally you can have. So for cranial chordomas, they happen in the brain, in the brain, ideally in the cerebrum. So we have cranial chordomas, and then the other one is not called ideally spinal; it's called sacral chordoma. So these cranial chordomas and spinal chordomas are coming across, first of all, due to um, this notochord, a failure of degeneration of the notochord. So they'll be larger than the cyst. And then for these chordomas, they won't form majorly in the nervous tissue. They can be absent in the nervous tissue. They can just form like just next to the nervous tissue. Just figure out the location of the notochord in relation to the neurotube. And then now you can be able to talk about the location of this. So for the chordomas, they form maybe outside the nervous tissue. Then for the cyst, the collection of cerebrospinal fluid um, in the nervous tissue at this point. Yeah, I presume with that we can call it a day. So just to encourage us that you just continue working hard, everything will be well. I'll see you maybe when you see me. I'll see you when you see me, and have a blessed evening ahead. Yeah, maybe to just remind us, these recordings will be posted in the YouTube channel. So you can go ahead and rewatch them if you feel like maybe you didn't get anything from it. And thank you for the support. Thank you for the support that you've shown. I really do appreciate for that. So maybe I'll see you later. I'll see you later. You guys have a wonderful time ahead.